Welcome to the Longevity Papers podcast.
Great to be here, ready to get into some potentially really interesting new science.
Absolutely. And just before we dive in, a quick note for you, our listener. Transparency is, well, it's crucial in science and how we discuss it. Today's paper comes from Shift Bioscience. Now, Shift is a portfolio company of HealthSpan Capital, which is a fund that the creator of this podcast actually co-founded.
Right.
So we want you to have that context. We're definitely going to be digging into the paper's findings rigorously, but, full disclosure up front.
Good to get that out there. Context is always important.
Okay, so let's get to it. Today's deep dive is on a brand new preprint, just landed on BioCurve, Friday, June 6th, 2025.
Yep, hot off the presses. The title is A Single Factor for Safer Cellular Rejuvenation.
And the authors.
It's a team from Shift Bioscience Ltd., folks like Delima Camillo, Gam, Maskalenka, and several others listed.
Okay, so the big claim, the headline that really grabs you, is about this thing they're calling SB00.
Right, a single gene.
A single gene intervention, they say, can rejuvenate cells from, well, multiple different origins, multiple germ layers.
And crucially, they claim it does this with efficacy that's pretty similar to the Yamanaka factors, the sort of gold standard we always talk about.
There's always a but, isn't there?
Well, this time it's a good but, potentially.
Yeah.
The critical twist is they claim SB1000 works without activating those really dangerous pluripotency pathways.
Okay, let's pause there. That difference, rejuvenation versus pluripotency, that's kind of the whole ball game right now in therapeutic development, isn't it?
Absolutely. The Yamanaka factors, OSK, sometimes OSKM if you include mymophantids, they were just monumental.
Totally changed the game. Induced pluripotency, turning any cell back into a stem cell.
Exactly. And along the way, it turned out they could also rewind the epigenetic clock, make cells look younger, even, extend lifespan in some mouse models of aging.
So they are the benchmark for age reversal at the cellular level, no question.
No question. Powerful stuff. But, and this is what the paper highlights right away, there's a sort of fundamental issue if you want to use them therapeutically for aging itself.
Because they weren't designed for rejuvenation?
Precisely. They were found and optimized specifically to induce pluripotency, to make stem cells. The rejuvenation was almost like a happy side effect.
And the problem with inducing pluripotency, if you just want younger cells, is safety.
Massive safety liabilities. The paper lays them out clearly. First, if you just keep expressing these factors constantly in an animal, lethal.
Lethal? Why?
Because specialized cells lose their identity. Your liver cells forget their liver cells. Your gut cells forget their job. The organs just fail. You need cells to know who they are.
Okay, so continuous expression is that. What about just like pulsing it, short bursts?
That's what people try. But even transient expression carries what they call teratogenic risk, basically the risk of forming teratomas.
Those sound bad.
They are. Tumors, but like a jumbled mess of different tissue types, hair, teeth, muscle, all mixed up. The paper calls it a body without a body plan. Chilling phrase.
Wow.
And the really scary part is they suggest even one single cell accidentally getting fully reprogrammed could potentially start one of these.
So you're trying to rejuvenate, but the tool keeps wanting to overshoot into this dangerous stem cell state?
Exactly. It creates this incredibly narrow therapeutic window, trying to get just the right amount of rejuvenation without triggering full pluripotency or losing identity or risking tumors. It's just extremely difficult.
What about leaving out See My Luck? That's the oncogene, right? Does OSK alone solve it?
They address that. And no, not really. OSK alone is still perfectly capable of inducing pluripotency and forming those stem cell colonies in a dish. The risk might be lower than with OSKM, maybe, but that fundamental danger is still there.
Okay, so the paper makes a strong case that the standard approach, the pluripotency link rejuvenation, is just inherently flawed for translation into therapies.
Yeah, it sets the stage perfectly. It basically says, we need a different way.
Which brings us back to Shift Bioscience and their approach.
Exactly. Their whole paradigm was different. They called it rejuvenation-first. Instead of looking for factors that make stem cells, they screened directly for factors that reverse cellular age.
How do you screen for age reversal directly? You need a tool for that.
Right. And that's their AC3 clock, mentioned in Figure 1A. It's their own proprietary tool.
Okay, proprietary. What kind of clock is it?
It's a single-cell transcriptomic clock. So it doesn't look at DNA methylation like many clocks we talk about. It looks at the pattern of gene expression, the transcriptome, in individual cells.
And uses that pattern to predict the cell's age.
Correct. They built it on a big data set of human skin fibroblasts, lots of different ages, different ancestries. And they did validate it against a well-known DNA methylation clock, showing good correlation.
What's the big deal about it being single cell?
That's key for screening. They claim it's the first truly single-cell clock, meaning you don't need to average data from groups of cells. You get an age prediction for every single cell.
Ah, so each cell becomes its own little experiment.
Exactly, which leads into their high-throughput screen, shown in figures 1b to 1d.
Okay, how did that work?
They took old human skin cells, fibroblasts, and used lentiviruses, basically delivery vehicles, to make cells overexpress one specific gene candidate at a time. They tested about 1,500 different candidates this way.
One gene per cell across thousands of cells for each gene.
Yep. Then after 2 weeks, they did single-cell RNA sequencing on hundreds of thousands of these cells to read out their transcriptomes.
And then applied the AC3 clock to each cell's transcriptome.
Precisely. To see which gene candidates made the cells look younger, according to AC3.
And the results, figure 1D.
Yeah, it shows the spread. Most of the 1,500 genes did basically nothing to the AC3 age. A few known factors had, modest effects, but then there was one clear outlier, SP1000. It showed a really pronounced rejuvenation effect, significantly reducing the AC3 age.
How did it stack up against the Yamanaka factors in that same screen?
In this specific screen, using their AC3 clock, the effect size they saw for SP1000 was comparable, maybe even a touch better than the full OSKM cocktail.
Okay, that's a striking result from a screen. But screens. This is where we need to be a bit cautious, right?
Absolutely. Standard scientific skepticism kicks in. First, that AC3 clock, it's proprietary, a black box.
We don't know exactly what genes it's using or how the algorithm weighs them.
Right. So could SP1000 just be really good at tweaking whatever specific signature that clock measures without being a true broad rejuvenator? It's a fair question at the discovery stage.
And screens themselves can throw up false positives or something called the winner's curse.
Exactly. Where the top hit in a noisy screen looks maybe better than it actually is in follow-up tests.
But it seems like the authors knew this criticism would come.
Definitely. The paper structure shows that. They immediately pivot from this proprietary screen finding to extensive validation using public, widely accepted methods. That's crucial.
So, figure 2. This is the validation part. Testing SP1000 more rigorously in aged fibroblasts, both skin and lung cells.
Correct. Lots of experiments here. First, they looked at the transcriptome again, but with more standard analyses.
Did the AC3 age reversal hold up?
It did. They confirmed a significant reduction, about 4.5 years mean AC3 age reversal, which was again comparable to OSKM in these validation experiments. But they went further.
Looking at things like senescence, that's figure 2E.
Yep, using the Simeo score. Senescence is that state where cells stop dividing but hang around, causing trouble.
What did SP1000 do?
It significantly reduced the senescence score across the whole population of cells. But the way it did it was interesting.
How so?
It was a homogeneous effect. The whole distribution of cells shifted towards less senescence. OSKM, in contrast, had a more heterogeneous effect. With OSKM, some cells got less senescent, but others seemed to get pushed into an even more deeply senescent state. It suggests OSKM might be causing some kind of cell fate split, pushing cells in different directions.
Whereas SP1000 gave a more uniform benefit, like dialing back the whole population together.
That seems to be the implication, yeah. A potentially safer profile in that sense. They also looked at broader aging hallmark pathways.
What did they find there?
SP1000 seemed to beneficially tweak 11 out of 20 pathways, stuff related to the cell's structure, inflammation, protein, quality control. OSKM also hit 10 beneficially, but it negatively impacted 8 others, including pathways linked to DNA instability and chromatin changes.
Those sound like the kind of pathways you don't want to mess with too much if you're aiming for safe rejuvenation.
Exactly. Classic reprogramming-associated downsides that SP1000 seemed to avoid, at least at the transcriptomic level.
Okay, transcriptome looks promising, but gene expression can be temporary. What about the more stable epigenetic changes? DNA methylation. That's figures 2G to M.
Right, this is arguably even more compelling data. They looked at methylation patterns after 6 weeks, giving things time to stabilize.
And the key finding was figure 2I, global methylation.
Yes. So generally, as cells age, they tend to lose methylation globally across the genome. It's a common aging signature.
What did SP1000 do to that?
It did the opposite of aging. It increased global CpG methylation by about 3%, pushing it back towards a younger state.
And OSK?
The OSK did the opposite again. It caused a significant decrease in global methylation by over 2%. And that global demethylation, That's a known hallmark of pluripotency induction, wiping the slate clean.
Wow, okay, that's a stark difference. They're having fundamentally opposing effects on this global epigenetic mark.
It's a really strong hint that they're working through very different mechanisms. And this difference carried through beautifully to the epigenetic clock results in Figure 2K.
These are the standard public clocks.
Yes, clocks validated in large human studies to predict age, health, even mortality risk, like Horvath's clock, Skin and Blood, Grimmage 2, Dundon Pace.
And how did SB1000 perform on those?
Remarkably well, often better than OSK in the same experiment. For the Horvath 2013 clock, SB1000 reversed age by over 7 years. OSK was just over 3.
7 years versus 3, that's a big gap.
It is. For the Skin and Blood clock, it was over 5 years for SB1000 versus less than 1.5 for OSK. Grimmage 2 reversal was similar between them. And Dundon Pace, which measures the rate of aging, was also significantly slowed by both, maybe slightly more by OSK in that case.
But overall, SB1000 wasn't just matching OSK on these public clocks, it was often beating it, sometimes substantially.
Correct. And supplementary figure S2 shows this held up across like more than 20 different epigenetic clocks. It wasn't just cherry-picking one or two.
So how do they interpret this? Why would SB1000 increase global methylation and reverse clocks so well while OSK decreases it?
The thinking, hinted at in the discussion and some supplementary figures looking at specific gene sites, is that SB1000 might be promoting some kind of identity-preserving repair. It's fixing specific age-related epigenetic damage, particularly maybe hypermethylating certain sites that should be silenced while leaving the cell's core identity program intact.
While OSK is doing more of a global wipe.
Kind of like a demolish and rebuild approach,
Yeah.
driving towards that ground state of pluripotency, which involves widespread demethylation. SB1000 seems more targeted, more like careful restoration.
And that mechanistic difference is absolutely central to their big claim. Safety.
Yeah.
Decoupling a rejuvenation from pluripotency. That's figure 3, right?
Exactly. Figure 3 is all about demonstrating that safety aspect. Can it rejuvenate without the risks? First, they checked if SB1000 messed with the cell's identity.
Figures 3A to C.
Yeah. They used markers for fibroblasts. SB1000 barely disturbed the fibroblast signature compared to control cells. OSK and OSKM, as you'd expect, caused a major loss of that signature.
They also used a cell classification tool.
Right, SCAB. Based on the transcriptome, about 70% of SB1000 cells were still confidently ID'd as fibroblasts, same as controls. For OSK, it was only about 42%. The rest were ambiguous or maybe heading somewhere else.
So the SB1000 cells still basically knew they were fibroblasts.
It seemed like it. And then figures 3D to F visualize this trajectory idea. They use pseudotime analysis. Think of it like ordering cells along a path from being a differentiated fibroblast towards being a pluripotent stem cell.
Where did the SB1000 cells land on that path?
They stayed put, clustered right with the control fibroblasts at the differentiated end. OSK and OSKM cells, however, clearly moved along that path towards the iPSC state.
And figure 3F puts it all together.
Yes, that's kind of the money shot. It plots age reversal, how much younger the clocks say they are, against pluripotency progression, how far along that path they've moved.
What's the picture?
It shows SB1000 achieving age reversal comparable to OSKM, but with basically zero movement towards pluripotency. OSKM shows similar age reversal, but significant movement towards pluripotency. It's a stark visual separation.
That graph really drives home the decoupling claim.
It absolutely does. But the ultimate test for pluripotency isn't just molecular signatures, it's function. Can they actually form stem cell colonies?
The colony formation assay. Figures 3G to K.
Exactly. They put the cells in conditions where pluripotent stem cells thrive and form colonies. After 4 weeks, OSK and OSKM colonies formed.
SB1000?
Zero. None whatsoever. Definitive functional evidence, at least in this assay, that it's not inducing pluripotency.
Okay, no pluripotency. But did the cells still work like fibroblasts? They checked collagen secretion?
Yes, figure 3K. Collagen production is a key fibroblast job, and it typically declines with age. Induced pluripotent stem cells, iPSCs, make almost none. OSK cells made similar amounts to the age controls. But the SB1000 cells,
What happened?
they secreted significantly more collagen than the age controls.
Whoa, so it didn't just preserve function, it actually seemed to enhance it, boosted a youthful function.
That's what the data suggests. It really strengthens the case that this is rejuvenation within the cell's identity, maybe even making them more robustly functional, not just less old on a clock. The whole package in Figure 3 makes a compelling argument for a distinct, safer mechanism.
Okay, very convincing data package in fibroblasts. Mesoderm origin. But the big question for any potential therapy is, does it work elsewhere? Is it generalizable? That's figure 4, looking at keratinocytes.
Right. Skin cells, ectoderm origin, a completely different lineage. This is a crucial test. If it only worked in fibroblasts, its utility would be limited.
And what did they find in keratinocytes?
The results were, if anything, even more dramatic. They looked at epigenetic rejuvenation again after 6 weeks.
Same pattern with global methylation.
Yes, exactly the same pattern. SB1000 increased global methylation by about 4%. OSK decreased it by about 1.5%. That fundamental mechanistic difference seems conserved.
And the epigenetic clocks.
This is where it got really eye-popping. Figure 4E. The P.C. Horvath 2013 clock showed a median age reduction of, wait for it, 13.6 years.
13.6 years. In 6 weeks. How do you even interpret that?
It's a massive number. It suggests an incredibly potent effect in this specific cell type under these culture conditions, at least as measured by this clock. Dunin pace was also slowed significantly, by about 25%. And again, supplementary figure S7 shows it was consistent across dozens of clocks.
Wild numbers. What else did they learn from keratinocytes? Pathway analysis.
Yeah, they saw hypermethylation, suggesting suppression of pro-aging inflammatory pathways, things like TNF-alpha, interferon signaling. It's that inflammaging idea.
Connecting it to inflammation control.
Right. And using a predictive tool based on methylation, CpGPT, they predicted decreases in circulating inflammatory markers like IL-6. Interestingly, the pattern of these predicted changes correlated really well between the fibroblasts and the keratinocytes.
Suggesting a common anti-inflammatory effect across cell types.
That's the idea, a conserved mechanism. And importantly, the keratinocytes maintain their identity too, clustering correctly with reference skin samples based on methylation.
So, strong efficacy in 2 different germ layers, mesoderm and ectoderm, with conserved mechanistic clues like global hypermethylation and anti-inflammatory effects. That really bolsters the case for SB1000 targeting something fundamental in aging.
It does. The fact it was even stronger in keratinocytes is intriguing, not something you necessarily predict. Table 1 in the paper does a good job summarizing these key consistent findings across both cell types.
Okay, the data presented is undoubtedly exciting, very provocative. But now let's put our critical thinking caps firmly back on. Any major claim needs scrutiny. What are the caveats here?
Well, the first and most obvious one. It's a preprint.
Right, not peer-reviewed yet. Needs independent eyes, independent validation, standard scientific process.
Absolutely. These findings are preliminary until they pass that hurdle.
What about specifics to this paper? The big elephant in the room.
The anonymous gene, SB1000. We don't know what it is.
Which is understandable from a commercial IP perspective for Shift Bioscience.
Totally. But for the progress of science, it's a major roadblock. Other labs can't easily verify the findings if they don't know what factor to test. We can't look up its known biology, its normal function. It prevents the community from building on this work directly or understanding the context. Is it a known aging gene? A developmental factor? Something completely unexpected we just can't know.
It definitely slows things down for everyone else.
Massively. Then there's the echo of the screening caveats.
Even with the validation.
Well, the validation is strong using public methods. But we should still remember the discovery came from that proprietary AC3 clock and a high-throughput screen. Is it possible the real effect size in a broader context might be slightly less dramatic than the initial winner suggested? Maybe. Is it possible the AC3 clock had some bias that SB1000 happened to counter perfectly? Less likely given the public clock validation, but still a background thought.
And those huge effect sizes on the clocks, negative 13.6 years in 6 weeks, can we really take that number literally?
That's a really important point of interpretation. These clocks were trained on decades of slow, in vivo human aging, applying them to rapid, artificial, in vitro changes induced by gene overexpression. It's a different context. The clocks are clearly detecting a massive biological shift in a useful direction, which is significant. But translating that precise number of years reversed directly to potential human lifespan extension from this data alone, that's a leap. The experimental system itself could introduce artifacts affecting the readings.
And the biggest limitation inherent to almost all early-stage work like this.
It's all in a dish. Cell culture.
Flat plastics, single cell types, artificial media.
Exactly. It lacks the entire complexity of a living organism.
Yeah.
No tissue architecture, no cell communication between different types, no immune system, no systemic hormones, no mechanical stress. Showing something works beautifully in 2D monoculture is a fantastic start, an essential first step. But it is not the same as showing it works safely and effectively in a whole, complex, aging animal.
Which leads directly to what's next? What absolutely has to happen for this to move forward?
The paper outlines it well, and it's pretty clear. First, they need to dig into the mechanism. How does SB1000 actually work?
Even without revealing the gene publicly, they can do that internally, right?
Oh yeah, they know the sequence. They need to do the omic storm, transcriptomics, proteomics to see what pathways change. Use functional genomics like CRISPR screens or RNAi to knock down downstream targets and see if the rejuvenation stops. Figure out if SB1000 directly binds DNA or interacts with key proteins using things like ChIP-seq or co-IP. Understanding how it works is vital.
Builds confidence, might reveal other targets.
Exactly. They also need deeper in vitro work. More functional assays in fibroblasts, maybe wound healing, stress resistance. For keratinocytes, maybe testing in 3D skin models for more complex. And crucially, long-term safety and culture. Keep these cells growing for months and months. Do they stay stable? Any signs of genomic weirdness or transformation risk emerging over time?
But the absolute make-or-break experiments, the ones everyone will be waiting for.
In vivo. Got to go into animals.
Which animals?
Aged, normal, wild-type mice are key. They represent natural aging. Progeroid models that age super fast can be useful too, but you really need to show it works in normal aging.
And delivery. How would you get SB1000 into a mouse?
Probably AAV vectors. Adeno-associated virus. It's a common, relatively safe gene therapy delivery tool. Could be systemic, could be targeted to specific tissues.
What do you need to see in those mice to be convinced?
The primary goal has to be improved health span. Does it make old mice functionally younger? Better grip strength, better coordination, better memory, less frailty. Those are the key readouts. Lifespan extension would be fantastic, but improved health span is the immediate target.
And confirming the molecular changes.
Yes, you'd want to show epigenetic clock reversal in multiple tissues in the mouse. Does it actually work systemically?
And the absolute #1 priority in vivo.
Safety, 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 especially given the history with Yamanaka factors. Long-term, rigorous monitoring. Are there any tumors, any organ toxicity, any weird immune reactions? This is where OSK-based approaches have really struggled in translation. SB1001 has to clear this bar convincingly in animals.
They showed mesoderm and ectoderm. What about the third germ layer?
Endoderm. Good point. To really claim broad applicability, they need to show it works there too. The paper suggests this. Take stem cells, make them into, say, liver cells or pancreatic beta cells in a dish. Those are endoderm derivatives. Treat them with SB1000. Do they get younger epigenetically? Do they maintain or improve their specific function, like making liver proteins or secreting insulin? Success there would really cement the idea of a universal mechanism.
Okay, so wrapping this all up, what we have here is a preprint from Shift Bioscience presenting some really quite exciting in vitro data for this single factor, SB1000.
Yeah, the evidence suggests it rejuvenates cells from different origins, performs as well or even better than Yamanaka factors on key epigenetic clocks.
But the key claim is it does this without inducing pluripotency, maintaining cell identity, and potentially enhancing function. That safety angle is the crux.
It is, but we absolutely have to temper the excitement with the caveats. It's a preprint, needs peer review. The gene identity is secret, hindering wider scientific progress right now. And critically, all this data is from cells in a dish.
The real test, the proof, has to come from rigorous in vivo studies showing both efficacy, improved health span, and long-term safety in animals. Those studies are yet to come.
Exactly. Which leaves us with the big, tantalizing question this paper poses. If SB1000A, or something like it discovered this way, can prove safe and effective in a living organism, what does that mean? Could we actually have therapies that turn back the clock on cellular aging without the inherent dangers of stem cell reprogramming?
It hints at a whole new therapeutic paradigm, doesn't it? Focusing on repair and rejuvenation within a cell's specialized role, not by erasing it.
The potential is just enormous if it pans out. But yeah, that journey from petri dish to patient is incredibly long, incredibly difficult.
We'll definitely be keeping a very close eye out for those follow-up studies, especially the in vivo results. Fascinating stuff.